will not guarantee for the uniform dispersion of the nanofiller into the composites. Into the matrix forming nanocomposites. So that way, shearing technique is not a very popular technique or a very convincing technique to form the nanocomposites, particularly if you go for elastomers, right? That is the rubber-based nanocomposites. So what is being done in case of this nanocomposite fabrication is that intercalation and exfoliation techniques, whereby through polymerization, in situ polymerization with the monomers, the exfoliation or intercalation of the layered nanoparticles, they are being tried to disperse within the polymer matrix. And as and in situ polymerization as such, where we can polymerize in case of say nanofibers, that monomer to polymer in presence of that nanofibers, that in situ polymerization. Then polymerization in constrained spaces. If you have some porous kind of nanoparticles, then you can have that constrained space polymerization of the monomers. And also this polymerization around these nanofibers, nanotubes, through that in situ technique or interfacial technique, polymerization technique. So these are the ways by which we generally try to synthesize these nanocomposites, because what you need to have a uniform dispersion of the nanofiller within the polymer matrix. Otherwise, whatever you are intending to have in from the property point of view, you will not be able to get that. Now, once you have this nanocomposite, so you are actually making some functional materials out of these structural materials, right? So macro composites, which are exclusively the structural materials, and when you are getting into this nanocomposite domain, so apart from that structural functioning, it can also give you some specialized functioning, some specialized activities. So you are actually what you can do, you are doing the, what you can say, that the value addition you are doing when you are forming these nanocomposites. Next slide, please. So, as I mentioned that these nanofillers, at least in one dimension, it has to be nano when you are referring it as a polymer nanocomposites. So, what we call it as a one-dimensional nanocomposites, where you have these nanofillers in lengthwise, they are not nano, but in diameter-wise, they are nano. So, these rigid rod type fillers, when you are using it, for the formation of the nanocomposites, then you can call it as a 1D nanocomposites, and that is from the perspective of the filler dimension. Then you can have 2D, where you can have both the width and thickness. These are within the nano dimension, where you are using clays, mica, aluminum, silicates, that is the plate type fillers. And these we refer as the 2D nanocomposites. And 3D or zero dimensional nanocomposites, where you are using some conventional or unconventional inorganic fillers. Conventional means when you're using some metal oxides, say like silica, zinc oxide, these sort of fillers. So these are conventional fillers we know. And when you are using them in the nanoform, and you are trying to disperse them within the polymer matrix, and unconventional means where when you are going for say silver nanoparticles, when you are going for say gold nanoparticles. So these sort of nanoparticles, when you are trying to use or trying to disperse within the polymer matrix, and those metals, metal oxides, these sort of nanoparticles, what we consider, they're having a spherical shape. And when you are considering a sphere, then it is a nano from all dimensions. So that way, what we call it as a three-dimensional nanoparticles and 3D nanocomposites that we are forming using this nanomaterials. So eventually, in a polymer nanocomposites, actually we are getting hybrid formation between this polymer matrix and these sort of nanomaterials, nanoparticles, and nanofibers. Next, please. Now, this fabrication of these nanocomposites, as I mentioned already, so here is once again a repetition of that, just to keep you informed that for these three-dimensional inorganic structures, when you are using or when you are trying to disperse, then what you should do, you should go for that polymerization inside or around a mesoporous or microporous solid or fibrous fillers. So that would give you a very effective dispersion of this sort of nano material within the polymer matrix. When you go for 2D nanocomposite synthesis, then you must go for this intercalative or exfoliative polymerization technique, where you should go for that in situ polymerization, which is most common, and some melt intercalation you can also try, particularly with the polymers which are of a lower molecular weight. The melt intercalation is more effective for those cases. And if you want to go for that zero dimensional, or what we call it as a 3D nanocomposite formation, then sol gel technique has been proved to be most effective technique for the nanocomposite synthesis. Now, these nanocomposites, depending on their method as well as conditions of synthesis or preparation, they actually exist in a metastable state, which is far from equilibrium. So that is why the to get a very stable nanocomposite is a very difficult job to handle, particularly aggregation of the nanofillers, because more often we see that the dispersion is not homogeneous. The dispersion is difficult to be stabilized, and the reason is that this is a metastable form of the material, and it will always try to equilibrate uh, or to achieve an equilibrium through that aggregation effect. So that nanocomposite synthesis, basically, although it is now almost 30 years old research domain, but still it's a challenge. And also there are two other key challenges that I'm going to mention. So as this is still a challenge to us to have a very stable and uniform nanocomposite formation from polymer using a polymer matrix. Next. Yeah. So one challenge that I have just mentioned, that is to stabilize it in its metastable form, and particularly because of this metastability, we have two other very important aspect to address, or what we call it as the unmet challenges of the polymer nanocomposites. And first is that the poor understanding of the nature of interface between the nanoparticle and the polymer, and that still exists. That is the surface modification of the nanoparticle, nanofiller, how often we do it to get a very good dispersion, a very uniform dispersion of the nanoparticles. So that compatibility and the stability issue of the modifiers to the polymer processing conditions, their stabilization, and the ability of the polymer to weight that surface modified nanofillers and all. So that is actually is what we can say that still difficult to assess, and to have a very generalized process or a very generalized rule to stabilize the system, particularly when you are processing it at a high temperature. Because when the polymer flow starts to flow during that processing temperature, the morphology actually is changed, right? So that is an important aspect of the polymer, and that again originates from that metastability of the system. And the second the challenge is that inability to derive any generalized rules of behavior for nanocomposites, right? We just not being able to derive any generalized rule that how a nanocomposite would function. For example, say polypropylene clay nanocomposites, they show property enhancements, whereas polyethylene clay nanocomposites show almost no property improvement. right? So that is very difficult to judge, that is very difficult to understand, because both polypropylene and polyethylene, they are very close in their structure, hydrocarbon polymer, most common polyolefins what we call it. But in one case we see that nanocomposite is giving some significant improvement, whereas in other case it's not much improvement is achieved with the same nanopillar. Again, say with polypropylene, property enhancements are observed with higher MFI materials, that is, polymers showing higher melt flow index, that is, having higher flowability, whereas no enhancements are observed with lower MFI materials, that is, polypropylene grade having lower melt flow index. So that is again within the polymer itself, when you are varying the molecular weight from low to high molecular weight, right, when you are moving from low to high molecular weight, then you see that the amount of change that you are getting, say at a low molecular weight, that almost you are not achieving that at a high molecular weight. So it is just to demonstrate that how difficult it is to predict the enhancement. Then the relative roles of internal modifiers and externally added modifiers or compatibilizers are also inadequately understood till days. What compatibilizer could function the best, or what compatibilizer could, be very good for dispersion of the nanofiller and the improvement of the properties? There is also no logic behind that, or strict, rule behind that. And also another important aspect, that this addition of these nanoclays or other nanoparticles, what we have seen that they actually affect the crystallization of the semi-crystalline polymers. So they are not just an inert particles or inert fillers, so they have some active roles to play also. So these are very, very important aspects to investigate, uh, to look into it. And actually this poses the, a challenge as well as an inspiration for future research, the and also the keeps the scientist interested to investigate in this field, although it is 30 years old now. With these aspects of the nanocomposites, let me now tell you a little bit on the tethering the polymer structure, how that polymer structure can be played with to get these say nanopolymers, or to change the dimension of the polymer polymer molecules, to give some useful variation in properties. Next, please. So, architecture of the polymers, mostly the polymers, once we refer a polymer, we actually mean that we are considering a linear polymer. Now, linear polymers, in linear polymers, you can have, say, in the first row, that you can have a homopolymer, you can have a block copolymer, right? Then you can have a graft copolymer, you can have randomly graphed or statistical copolymer, or you can have a periodic copolymer or gradient copolymer. So these are the varieties that you can have in linear polymers which have two chain ends, right? And when we talk about these your polymers, we generally mean these linear polymers. Now, graph copolymers is a bit different from these other linear polymers, where you have these polymeric grafts hanging from the main chain. So here you can have some multiple chain ends, and if you see that a graft copolymer shows a completely different properties from these all sort of these linear polymers. So here you first notice that when we move away from linearity, then we can meet with some interesting properties of the these polymeric molecules. So they are actually, these what we can say, that the interest of moving aside from the linear structure to the branch structure, actually that originated. And here you can see so many branched structures that we can have in polymers, these branched architectures, where you can have the branched, what we call it as a hyperbranch structure. Then gel structure means where through branching you have the cross-linking among the branches that gives you a very insoluble and infusible mass, what we call it as a gel. So that sort of gel structure, hyperbranch structure. Then your site specific functional your branch structure. So these are various tailor made branch architecture you can have. And why am I referring this? Because while you are going for branching, then actually you reduce the molecular size of the polymer. A linear polymer can unfold and increase its hydrodynamic volume, whereas a branched polymer actually it tends to be more spherical. And you know that when a mass tries to become tries to go in a spherical dimension, actually it reduces its surface area and reduces the size. So that way, that when we go for branching and extensive branching in continuity, then actually we are gradually reducing the structure of the polymer, and we can come within that nano dimension, and very smaller nanoparticles, nanopolymers can be synthesized, while you can go for this sort of branching and hyper branching concept. Now, this is a different concept from that of the conventional polymer nanoparticles that we refer, which are again based on that linear polymers, right? Now, linear polymers, they are being synthesized in the form of nanocapsules, your nanospheres, using several techniques sometimes not very conducive to carry out industrially with higher yield. So that is why we are venturing into this, technique or this process, whereby just by simply modifying the architecture of the polymer, we can synthesize a very, very small nano-dimensional polymer particles. Next, please. So why I am stressing on that changing the architecture from linearity to the branched, because once you move into this sort of branched architecture, then you can change the chain entanglement density, right? The chain entanglement density can be severely changed. Rather, the polymers will be less entangled when it is branched as compared to when it is in the your linear dimension. Then molecular size, as I mentioned, that when it is more branched, then the molecular size will be reduced, severely reduced, and I will demonstrate this with the help of the data. I'm going to show you. Then glass transition temperatures. In fact, a highly branched or a hyperbranched polymer can have a series of glass transition temperatures, whereas the linear polymers, they have a particular glass transition temperature. When you are going for, say, block copolymers, then depending on the block length, you can have two or three glass transition temperatures, but otherwise it is mostly a single glass transition temperature for the polymer, linear polymers. But for this branched and hyper branched, you can have a hierarchy of glass transition temperatures depending on the branch length. Then mechanical properties, you will not gain in mechanical properties, that's for sure, when you go for branching, because you are reducing the chain entanglement density, and while you are reducing the chain entanglement density, then you are actually losing the mechanical property. So that is one negative aspect of this sort of architectural polymers. But no worry, because there are so many sophisticated applications where actually you don't require that much of mechanical properties, right? Your strength properties and all. But I can show you one example where you can have a combination of good mechanical property and this sort of architectural input. I will show you also. But in general, when you are synthesizing a branched polymer, right, from the monomers, there usually you do not gain much of the strength or mechanical property from that polymer. Then you can have a much modified solution and melt properties. In fact, you can have a very low viscosity of these polymers, branched polymers. So you can increase the solid content without much increase in viscosity of a solution. That is some sort of a unique behavior of the polymer and a contrasting behavior from that of the linear polymers. Because for linear polymers, if the concentration increases, the viscosity increases tremendously. So that way, the solid content you have to keep at a very low value in order to have a workable viscosity. But in case of these branched, hyperbranch polymers, you can have even 30-40% of the solid content without much rise in the solution viscosity. And also melting, these polymers are going to be, or these polymers are very low melting polymers. Flow modifiers, these sort of polymers. Uh, Previous slide, please. We have not completed yet. Yeah. Surface properties. Now, as you have more branches, then you have more chaining. So you have, much higher functionality. So you can have a very improved surface behavior of these polymers. And most importantly, many void spaces, intramolecular void spaces you can have, which is absent in case of linear polymers because they are so entangled that you won't find much void spaces. But these you will have a huge amount of void spaces in these sort of branched and hyper branched polymers. Next slide now. So when we talk about these branched and hyper branched polymers, then we must refer this polymer, and I think many of you have heard this polymer, which is called dendrimer, and this is an example of the perfectly branched polymer. Perfectly branched polymers. Now, these dendrimers, they are tree-like polymers having branching out from a central core and subdividing into hierarchical branching units, and for that they forms a very compact globular structure. The structure is absolutely symmetrical, and you see that when it is spherical and symmetrically branched, then the size is restricted within 15 nanometer despite having much higher molecular weight, right? Now, these are having very dense surface surrounding a relatively hollow core. The core is hollow, but as you have expanded branches at the surface, so the surface is dense, whereas the core is hollow. And they have a very high degree of surface functionality, and they have excellent solubility and low solution and melt viscosity, just as I referred, and enormous amount of free volume spaces in them, right? So these dendrimers actually are the perfect branched polymers having nano dimensions. So next slide, please. Now, when we talk about dendrimers, then we can have another, say, imperfectly organized, dendrimers, what we call it as a hyperbranch polymer. Now, what is the, reason that we are mentioning this hyperbranch polymer, or we are referring this hyperbranch polymer here because dendrimers is as you have seen in the previous slide that these are highly symmetrical. So when you have, you want to have that much of symmetry in the polymer structure in terms of branching, then you have to put a lot of effort in synthesizing it, right? So for that reason, the synthesis of dendrimer is a bit hectic process, requires a lot of control for to have a just exactly symmetrical structure. But if you can afford some sort of asymmetry, that is not exactly uniform branching, or much less organized branches, that sort of asymmetry, if you can afford, then the synthesis process becomes much easier, because here you are not required to have that much of control in the synthesis process. So these hyperbranch polymers is of that category of polymer where you can have unorganized branches, right? Unorganized branches, and that for that you have you do not require that much of stringent condition to synthesize it. So both of these hyperbranch polymers and dendrimers both are branched. One is symmetrical, another one is asymmetrical. So here we have a vis-a-vis comparison between this, and the first point is most significant, and that is both the polymers, because of their extensive branching, they show identical behavior. So here you see that symmetrical and unsymmetrical, that part becomes a secondary. Whether the branches are organized in symmetrical fashion or unsymmetrical fashion, that is not reflected in the properties of these two systems. So here you see that the I mean our job becomes much easier, right? Then for dendrimers, you have absolutely monodisperse, because all branches are symmetrical in all the molecules of dendrimers, whereas it is unsymmetrical and irregular architecture in hyperbranch polymers. In dendrimers, what we refer as the degree of branching is 1 because of absolute symmetry, and extensive branching. And since the hyperbranched structure, the branches are unorganized and they are less controlled, so we call it as a degree of branching less than 1. Now event distribution of functional groups you have in dendrimers, whereas here you have for hyperbranch polymer uneven distribution. Then as I mentioned, that you require some specialized sophisticated condition to have that much of control for dendrimer synthesis, whereas for hyperbranch polymer it is much easier to synthesis, and for that reason the polymers are cheaper. Now, as you require, what you can say is a strict control. Then, when there is some deviation in process parameters that affects the structure of a dendrimer, right? That affects the symmetry of the dendrimer. Whereas you can be quite casual while you are synthesizing a hyperbranch polymer. And dendrimers, they give a lower yield as compared to the hyperbranch polymer. So these are basically favoring points that why we can go for hyperbranch polymer uh, than dendrimer. So next slide. So these are the routes by which you can synthesize these hyperbranched polymers. One is your polycondensations for, the condensation route that we use for our linear polymers. So that same polycondensation you can use for these hyperbranching or this hyperbranch polymer synthesis. Then you can go for self-condensing vinyl polymerization route. Multi-branching ring opening polymerization route you can adopt, proton transfer polymerization route that you can also adopt. So these are the some specialized technique also along with this conventional technique you can use. Then you can go for A2B3 kind of double monomer methodology, that is particularly for that condensation route. You have to take A2, that is A is one functional group, B is another functional group. So one monomer containing two A groups and another monomer containing three B groups. So once you combine them in condensation, then you can go for the synthesis of the hyperbranch structure. And in vinyl polymerization category, you go for the Strathclyde methodology, that is the addition polymerization route to synthesize the hyperbranch polymers in the vinyl category. Now we have been, next slide, we have been working synthesis and its various applications since 2014. And we have adopted both the condensation route and this vinyl, polymerization route or Strathclyde methodology routes for the synthesis. And I will show you some results on that today. One in the area of nanocomposites, how we have synthesized, and another for the nanopolymers. So next slide, please. So till 2014, we have, that route or that kind of polymers we have synthesized for these applications. Now, what we call it is that from that conventional nanocomposite synthesis and the conventional linear polymer based, nanoparticle synthesis for various application, we are actually now moving into an era of architectural polymer based nanocomposites and nanopolymer synthesis and their various applications. Now, let me tell you that this hyper branching or this branched architecture in polymer, that synthesis and that kind of design or tailor-made polymer synthesis, these are not the recent invention. Way back in 1960s, PJ Flory, he has done a lot of kinetic study in condensation polymers that are having branches. But that was limited to that condensation, kinetic study in that time, and that was not explored beyond that. Now, once we are, started investigating in this nanoparticle, nanocomposites, nanomaterials, then these hyperbranch polymers or branching in polymers, this technology has once again evolved, right? And now we are talking about this more as compared to this linear polymer-based nanoparticle nanosphere synthesis and linear polymer based nanocomposites, because we have explored this for say 25 to 30 years, and we see a very little success till now in this area. So what we call it as a moving into the era of architectural polymer based nanopolymers and nanocomposite world. And in this era, what we are suspecting that the challenges would be that we are now trying to explore some unknowns with some little knowns, right? So that could be a challenge, and we need to have take up this challenge and see that how far we can be successful. And also the challenge is to go for some design of some user friendly technology which has the viability, because you know that after say 30 years of rigorous study on these various linear polymer based nanocomposites and nanoparticles, only some 10 to 15% success in commercialization we have achieved. So that is the the challenge also we need to take up in this technology, and we see that how much viable we can make this particular technology in large scale application. So this with this next slide please. So with this, as I mentioned that since 2014 we are working in this area. So in this field till now our contribution is about say 10 international publications we have so far. And we have authored this book, this hyperbranch polymers for biomedical applications. And in this book, we have in detail discussed about your synthesis and prospective applications of these hyperbranch polymers, and why we were taken up this hyperbranch polymer route instead of dendrimer, just as I have explained. And from that commercial viability angle or say easy preparation technique angle, these polymers are more advantageous compared to dendrimers. And in our point of view, that this is going to be the polymers in future for various sophisticated applications. Now, with this brief introduction about say polymer and nanocomposites from that conventional nanocomposite point of view, And moving into this era of say architectural polymer, I'm going to show some of my work, in fact my PhD scholars work, which we have done in this area. And next slide please. Yeah, and I would like to start with Tomalika's work, and to show you that how we have developed these hyperbranched grafts using conventional biopolymers, and also thereon we made the nanocomposites, and we have used it for the sensing of soluble mercury in water, right? And that too for the controlling the heavy metal pollution creating by the soluble mercury. So we have tried to develop a very cheap and very effective sensor for mercury using this hyperbranch graph technique, and subsequently following that technique, forming the nanocomposites. Next slide please. So first, what we have done is that we prepared the hyperbranch grafts from a biopolymer, and that biopolymer we have taken was carboxymethyl guar gum. Right now, carboxymethyl guar gum is a chemically modified guar gum, and that chemical modification imparts some higher solubility to the guar gum. Otherwise, guar gum is very lowly soluble in water, but your carboxymethyl guar gum, its solubility is much higher than that of guar gum. So we have started with this material carboxymethyl guar gum, and we have taken acrylic acid as a grafting monomer, and we have taken divinyl benzene as a brancher. Right. So this is the system we have started. Now I would explain that what is the background behind it. This technique was invented by your Sorensen in the year 1982, and he named this as a Strathclyde methodology for synthesizing hyperbranched vinyl polymers, where he designed the system in this way that he would use a monomer and a brancher and a chain transfer agent. Now, monomer must have a functionality of two, that is one unsaturation. A brancher must have functionality more than two, that is at least two unsaturation points, and a chain transfer agent. Now, the idea behind is that when there is a copolymerization between this monomer and the brancher, then the brancher would form the branches, right? And if the branching is allowed for long, then it will start to gel, right? So from the very beginning, a chain transfer agent is being used that will cut off the branch and allow the growth of the polymer in the branched mode. So that was a very simple concept proposed by Sorensen, and we tried to adopt that vinyl polymerization technique into grafting so that we can have a hyperbranched graft instead of a linear graft. Right. So that the idea we collated in our work, that we have taken preformed polymer like say carboxymethyl guar gum, and we have taken this monomer as a grafting monomer, and this brancher as a divinyl benzene, which has two unsaturation points. And we have taken dodecane-thiol as a chain transfer agent and SDS as a surfactant for this polymerization, and KPS potassium peroxide sulfate, that is ammonium potassium persulfate, as a initiator. And in presence of nitrogen, we carried out the polymerization to prepare these hyperbranch grafts onto carboxymethyl guar gum. Next slide, please. Yeah, these are the samples we have prepared. That is, first one, that is PAA1 that we have referred, is a linear graft, right? It's a linear graft because always you must have you should have a linear graft in this sort of work to see the changes, right? To record the changes or deviation, how much you are getting in the hyperbranch grade. So in order to measure or in order to have an idea about that, you must have this. So this first sample is the linear graft. Then the second one, that is PAA2, where we have used divinyl benzene 1 mole and 2 moles of the chain transfer agent, that is dodecane in twice of that of the brancher. And HBPA3, that refers to where you have 1:1 mole composition of divinyl benzene to dodecane, that is the brancher to your this chain transfer agent in equal molar proportion. And HBPPA4, where you have that more your brancher and less chain transfer agent, 1:0.5. And fifth one is that sample which is devoid of any chain transfer agent, only brancher is there, right? So these are the systems we have designed. So in the branching grade, basically, we have three samples: PA2, PA3, and PA4. PA1 is the linear one, and PA5 is the gelled sample that we are expecting, right? And we in fact got it in the form of gel, because we have not used any chain transfer agent. So finally, what we got is the gel, and you see that the many of the columns, the data are missing because the samples were highly insoluble. Now, these are the properties that we have measured to characterize this. First is the your carbonyl index, that how much your acid have been grafted. So here you see that in all the cases almost, we have almost close to 1 or greater than 1 value. So that indicates that the acrylic acid has been grafted successfully, maybe in the linear mode or in the branched mode. Then we measure the solubility. You see that the hyperbranched grades, they are having I mean your solubility, your this linear polymer at pH 6, it has the solubility of around 0.088. These hyperbranch polymers, among these three, that PA3, it is having higher, much higher, in fact, much higher solubility as compared to the other two hyperbranch grades and the linear grade. And we marked it with this red. And even at pH 8, among the hyperbranch grades, these three, that is where we have the 1:1 molar proportion of divinyl benzene to dodecane, it is showing the highest solubility among these branched or hyperbranched grades. Then we measure the intrinsic viscosity, and you see a drastic drop of intrinsic viscosity from the linear graft to that of the branched samples. And you see for the linear one, it is 1.5615, whereas in the branched grade, it is around zero point 0.1 only, right? So you see a very sharp change in the intrinsic viscosity values. And turbidity. Turbidity, you see linear polymer showing that 20 NTU Nephilo turbidity unit, whereas this HBPA3, it is showing the lowest turbidity of 8.2 NTU. So here we see that this PA3 is likely to be the best branched grade of sample, because it exhibited maximum solubility, minimum intrinsic viscosity, and minimum turbidity. Next, please. Now, these are the spectroscopic characterization. I am not going into the detail of it. I have, given here that what are the, characterizing peaks, both from these branched and the linear samples that we have, and also how we measured the carbonyl index from these spectroscopic figures. Next slide, please. This is from the NMR. We have characterized your presence of your brancher, presence of the chain transfer agent in the hyperbranch polymers, and thereby we can say that the branching has taken place in the your grafted chain. Next, please. So what we thought, based on the your spectroscopic characterization, the polymerization proceeded via ring opening of this polysaccharide at the beta carbon, and conversion of that gamma carbon into an aldehyde groups in the proton NMR and C13. We detected the aldehyde proton and that carbon, and that followed by that your graft reaction, whereby it is expected that we have the hyperbranched graft. But along with it, we also thought that we cannot avoid the formation of the linear grafts as well, because the reactions are random, right? The reactions are random in vinyl polymerization. So even then where the brancher or chain transfer agents are used, some percentage of linear graft are also expected to form in the hyperbranch grade. So basically, these hyperbranch polymers are, I mean, a combination of a major proportion of hyperbranch grafts with some proportion of linear grafts. So next slide, please. So this is the one of the most important characteristics of the branch polymers, that is the rheology. Because when you develop branches in a polymer and when you deviate from the linearity, then what happens? The polymer tends to be more Newtonian rather than pseudoplastic, right? So in the first figure, you see that the A sample, that is the linear graft, it has shows a pseudoplastic behavior, means a drop in viscosity with an increase in your frequency, whereas as the your branching is increased in the polymer, that is PAA3, that we marked in the red, that sample, it has shown minimum effect of frequency on its viscosity. So it indicates that it is most Newtonian as compared to other branch polymers, as well as from the linear graft. And also the second one, that top right, it shows the relaxation behavior study, where we see that only for the linear graft sample we record a crossover between the elastic and the viscous moduli, which indicates its pseudoplastic behavior and the relaxation characteristics. Whereas for the branch polymers, we do not record any such of your relaxation behavior. And the below figure, that is the tan delta, in the hyperbranched or the branched polymers, we record multiple peaks, particularly in that PAA3 sample, multiple tan delta peaks, which indicates that the your hierarchy of existence of that relaxation behavior in the chains, because of that multiple branches that we can have, that we had in this branched or hyperbranch structure. Next, please. Now, with these your hyperbranch graphs, now we try to synthesize the silver nanoparticles in order to develop that sensor, Mercury sensor. So what we did, we took these hyperbranched polymers. Also, we took the linear polymers as well, because again, we have to have a comparison between. So we took their solution, and we put silver nitrate in it, and in presence of the reducing agent sodium borohydride, we generated the nanosilver, and then we filter it to separate out the sample. Now, these silver nanoparticles generated in the linear graphs, when we used for that sensing action with mercury salt, we see that it is your first of all, the stability of that silver nanoparticles in the linear graph was poor, right? And we see that the blackening of the entire solution of that silver nanoparticle within a few hours. So first of all, first issue was the stability, and the second issue was that it was instantly making that mercury that solution black, right? So that was the second constant that we have with that linear graph. While we use that hyperbranched matrix for the formation of the silver nanoparticles, we found it very stable, and we measured the stability. In fact, we kept that solution for 6 months, and we found that it was not changing its color. We characterized it with the help of UV periodically in every month, and we have seen that the lambda max it was showing, initially it is not even changing more than 2 to 3 centimeter inverse. So that way, centimeter, sorry. So 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 that way we have studied the stability of that silver nanoparticles we generated in the hyperbranch matrix, and we are satisfied that this hyperbranch matrix it developed some highly stable silver nanoparticles as compared to the linear graft copolymer. Next slide, please. So here you see that UV characterization for our these hyperbranched and linear graph samples, and you see that the peak is varying between 436 to 450 for all the samples. And here the DLS study, and you see the size distribution, and that sample C you can see the most narrow size distribution, and much smaller within the range of around 65 nanometer of the average diameter, and it's a very narrow distribution you can see. So you can observe the first one, that is sample A in the linear grafted matrix, then B in the B, C, D are within the branched, and C is that that base sample we got. So it also gives you a very narrow distribution of the silver nanoparticles with a much smaller size. So next. So now we went for that mercury sensing study. Now we for any sensing application, you need to develop a calibration plot, and with that hyperbranch grade, we developed calibration of your this mercury sensing at different concentrations of the mercury, and we have plot this calibration this point into a calibration curve, and we see that these mercury, I mean this mercury solution, it is being decolorized your these silver nanoparticles, and but the your decoloration is not complete with these silver nanoparticles that is present in the matrix. So it is not completely decolorizing the mercury sample, but it is decolorizing to a certain extent, and we measured that your values at different concentration of mercury, and we plotted this calibration, and here is the TM picture of that I mean your mercury when there is a reaction between silver nanoparticle and the mercury. Now what we have seen when for this sensing application, that there are other samples also, like your zinc, then we can have your zinc, then your gold, these sort of samples, they are also decolorizing this mercury along along with this I mean these solutions are also getting decolorized with the silver nanoparticles along with the mercury. So our next job was to distinguish the mercury discoloration with the help of the silver nanoparticles, because otherwise we cannot have a very specific sensor, because if these ions are also present instead of mercury, they can also decolorize and there will be a wrong information. So next what we did, next slide please. Then next what we did, we tried this sensing reaction with the help of in presence of sodium chloride, right? And what we see that when sodium chloride is not I mean present, then we have this sort of reaction. We have given the visual, the silver nanoparticles that color some your reddish color, and when that mercury is added, mercury solution is added, after 2 minutes you get this sort of, as I mentioned, it was not a complete decoloration, so you have this kind of color developed, faint color developed, whereas if you do the reaction in presence of say sodium chloride, then you can have the complete decoloration of this silver nanoparticle compound or this your solution within 30 seconds. But importantly, the same reaction is not repeated, or the same reaction is not taking place when the calcium or gold, these sort of interfering ions are present. So that way we actually developed or being able to develop this sensor exclusive for mercury, right? So when we the idea was then that when there is a partial your discoloration of silver nanoparticles, then it could be due to mercury or calcium or gold. But if in second step the complete decoloration of silver nanoparticle does not take place in place of sodium chloride, then you can say that there were no mercury present, right? But if that decoloration in presence of sodium chloride takes place, then you say that the mercury is present. So that way we tried to develop an exclusive sensor, and that silver nanoparticles which remain stable for 6 months, so that also gives you a very good shelf life for using it. And with the help of the calibration curve, we were able to find out the mercury concentration even up to the ppb level. So that was what we can say, that with the help of this uh, your hyper branching architecture, we were successful dealing with this sensing application of these nano composites. Next slide, please. Now, these are the slides that tells you that why that silver, your sodium chloride was making that complete discoloration with the help of mercury. I'm not going into the detail of this. Next slide, we can skip this. Next one. This is again the XRD and the EDX proof. Yeah, let us come to the another work. Now, this is your on the nanopolymer synthesis within with this hyperbranched architecture. And here we have tried to develop some polymer which itself has a strong antimicrobial property. And can that be used as a nano drug in future? We need to see. So this work was done by Srijoni, whereby we have synthesized hyperbranched copolymers from polyethylene glycol and citric acid, right? Let's see how we made it. Next. So we have taken polyethylene glycol and citric acid at the two monomers. Now you can note that polyethylene glycol, it itself is a linear polymer having many hydroxyl groups, right? But still it acts as a bifunctional hydroxyl monomer in the reaction, right? So that is another beautiful thing that we achieved, that using your polyethylene glycol, we have been able to synthesize a polymer where the branching is not taking place from that polyethylene glycol point or polyethylene glycol part, right? Now, what we have done, we have taken polyethylene glycol or PEG of three different molecular weights. One is of 4,000, another 6,000, and the third one is 20,000. And the reason behind this is to vary the linearity, a linear portion in the branched architecture. Because citric acid is a trifunctional acid, so citric acid will act as a nodal point of branching, right? So that polyethylene glycol will give you the linearity in the chain. So while you increase the molecular weight of polyethylene glycol, so you are expected to have more linearity in the chain. So that was the idea behind taking three different molecular weights of polyethylene glycol. And what we have done, that we have your kept the mole ratio of them is 1:1, because even in the 1:1 mole composition, if polyethylene glycol is bifunctional, then citric acid is trifunctional, so it will give you a branching system, because the average functionality is more than 2. So we developed S1, S2, S3, where S1 was the sample we developed, or polymer we developed using the lowest molecular weight PEG, that is 4,000, S2 with 6,000, and S3 with 20,000. And here you see that yields were more than 70% in first two case, and for third one the yield is little bit lower, right, about 67%. And if you consider solubility, you can see that for S1, you have the highest solubility of about 96%, for S2 about 94%, and S3 about 92%. But overall, we have a very good amount of solubility for this all the three polymers. So next slide, please. Yeah, so we thought that we have this kind of architecture developed, this sort of branched architecture developed with your PEG and citric acid. S1, we expect more branching because the linearity is less, right? Linearity is less, so it is expected to have more branched architecture compared to S2 and S3. Next, please. And we determined the chemical structure of this from IR and NMR, and from NMR we tried to find out the percentage of branching by comparing the NMR peaks, and we found out that about 70% branching in S1, and that was the maximum, and for S2 and S3 the branching percentage were lower. Next. Yeah. So we measured the intrinsic viscosity, and we see that S1 it has the minimum viscosity, followed by S2 and S3, and this trend was probably because of the trend that followed in branching, that S1 having maximum branching followed by S2 and S3. And the right hand side figures, this shows the DLS size distribution, that is the hydrodynamic size distribution, and importantly you see the your bottom right one, that is for PEG 4000, that is the polyethylene glycol having a molecular weight 4000, and you see the size of this linear polymer, right, it is about 4000, right, 4000. Whereas when you compare or when you see the sizes of these copolymers, right, it is within 4 to 5 nanometer. So you can see a drastic change in size that I was mentioning in the in my earlier part of discussion, that when you induce branching, then the size reduces drastically, and that too in the range of 2 to 3 nanometer even, and that what we have recorded in case of S1 having a maximum branching, and its size is about 2 to 3 nanometer, right? So this way it's a clear demonstration that when you can form branches, then you can reduce the size drastically, right? So uh, so what we next measured is the size at different pH, pH 3, pH 7, and pH 9. And the reason behind this measurement is to see if the change in pH changes the size of the polymer, because you have these acids present, and beyond the pKa value of this acid, which is around 5, which is around pH 5. So this acid will turn into CO2 minus, and because of the repulsive interaction between CO2 minus, had those acids present in the branch ends, there will be an expansion in size. So whether we are getting an expansion in size at a pK, I mean beyond the pK value of this acid, we wanted to check, and we see that there is hardly any difference in size at I mean above and below the pK value of the this acid, citric acid. So that way we confirm that the ends are probably locked with your PEG molecules instead of citric acid molecules. So the citric acids, they were present in the internal part, and the branches were ended with the PEG or that is polyethylene glycol part. So that way the polymer becomes insensitive towards the change in pH, the size particularly. Next. So here is the rheological study. As I mentioned, the rheological study is one of the most convincing way to characterize the branching, because while you go for branching, the your mechanical strength will go down because you are losing the cohesive interaction and entanglements, but at the same time the flowability or Newtonian character will improve. So what we see that this Newtonian character is maximum in S1 followed by S2 and S3, and from the size or modulus perspective, sorry, strength perspective or modulus perspective, we see the S1 is having the lowest modulus followed by S2 and S3. So that again characterizes that the branching, existence of branching and maximum branching in S1. Next, please. So with this S1 having this maximum branching, we went for these your antimicrobial property both with gram positive and gram negative bacteria. So we have taken B subtilis as the gram positive and E. coli as the gram negative bacteria, and we can see at the doses of 0.5 and maximum 1% concentration of this polymer, we see a severe, inhibition in the growth kinetics for both the bacteria. So that was an interesting observation. Generally, polymers do not act simultaneously in gram positive and gram negative bacteria. Not only polymers, most of the antimicrobial agents, not simultaneously on the both types of bacteria. But here we have been able to synthesize a polymer which was equally active in both gram positive and gram negative bacteria. Next, please. So we studied the cell viability, and we have seen about 96% viability at 0.5% concentration, and about 92% viability with 1% concentration of S1 with raw 264.7 cells. Whereas for S2 and S3, the results were also equivalent. So that way, this is another important observation that our polymers are not toxic for other cells apart from those gram positive and gram negative bacteria. And also we studied the Ross generation, and we found that the controlled Ross generation in case of S1 more controlled as compared to S2 and S3. And that was again an important observation and characteristics with this polymer. And what we suspected that the PEG, as these are present in the end part of these chains, being PEG having a strong antioxidant property, that induced the Ross generation in these your cells, and that actually the advantage we got with this PEG in this copolymer. So next slide, please. So there, this is your antibacterial efficacy study, and we measured the these your cells which are dead and which are alive with these your polymers S1, S2, S3, and the observations are, next slide, please. That both your these E. coli and B. subtilis, they demonstrates the number of colony forming units, and it was demonstrated a sharp positive impact of branching on killing both gram negative and positive bacteria, that already demonstrated through the plates. And S1 with the lower molecular size and maximum number of functional chain ends that containing PAG at the chain ends, they were capable of diffusing the bacterial cells much faster and efficiently, and that is why we got the maximum killing efficiency with S1 at equivalent dose as compared to S2 and S3. So we can see the direct effect of having much lower size of this sort of antimicrobial polymers in their functional efficiency. So now we can move to another work. Next slide, please. And here I would like to show the this use of these hyperbranched copolymers as a 2D scaffold for tissue engineering. Now, these polymers we have synthesized from citric acid and polyvinyl alcohol. So in place of PEG, we have taken polyvinyl alcohol. Now, polyvinyl alcohol, the reason for taking polyvinyl alcohol is that its biocompatibility. Here we are trying to have a different application from just what we have mentioned. So we need a strong biocompatibility in order to develop the cells immobilization and their growth. So for that we synthesized copolymers from citric acid and polyvinyl alcohol. Citric acid being a trifunctional acid again acts as a nodal point for the synthesis of the branches. Now let's see how we made it. Again, condensation route. Next slide, please. So we adopted condensation route and we have taken polyvinyl alcohol citric acid. And here what we have done, we have increased the concentration of citric acid in order to have less to more branches by keeping the polyvinyl alcohol at the same molar concentration. The molecular weight of polyvinyl alcohol was 1,15,000, and the polymerization was carried out at 135 degrees Celsius for 6 hours. So these polymerizations, the previous one, this condensation route, what we have done, we have just mixed it, maintained the temperature, put it on stirring for 6 hours, right? Just this is the way we made it, right? So this as I was mentioning that the hyper branching is much easier to make. So this is the demonstration of that how easily you can make these polymers just by mentioning maintaining the temperature and stirring. Now, these polymers, as I mentioned, that we varied the proportion of citric acid from low to high in order to have more branches, and the samples were V1 having lowest citric acid, V2 intermediate, and V3 highest. So it is expected that V3 will give me highest amount of branching, V2 intermediate, and V1 is the lowest. And here we have the yield of about 70, more than 70%, which is quite good. And we see the solubility where we have the more branches, V3 the solubility maximum 96%, and V1 minimum solubility little bit lower, about 92%. But overall again we have the very high amount of solubility of these copolymers, branched copolymer. Next please. Yeah, so what we see that in the schematic we have shown this V3 having the maximum branching and V1 the minimum branching, and we have your measured the intrinsic viscosity. You see, we compared with that of PVA as well. PVA being a linear polymer having 1,15,000. You see what is the tremendous amount of intrinsic viscosity it has shown, whereas we have seen a drastic drop in intrinsic viscosity from that of linear PVA to the branched polymers, and minimum value with V3 having maximum branching. Next, please. So this is the characterization, spectroscopic characterization, FTIR and NMR. For all these hyperbranch polymers that we have synthesized, we performed NMR in order to characterize them as well as to find the percentage of branching by comparing the peak heights. So what we found that in V3, the percentage branching was 72%, and for V1, V2, the branching was lower. In V1, it's about 30% that we have measured. Next slide, please. Yeah, so this is again the DLS size, that is the size is measured in dynamic light scattering. So for comparison and to demonstrate the sharp change in size, we measured the size of the polyvinyl alcohol as well, and you see it has given your bimodal distribution. It has given a bimodal distribution, and you see that the minimum size range it has shown is about 1,000 nanometer, right? Whereas when you go for this V1, V2, V3, you can see for your V3, the size is within your 10 nanometer, and V1 where we have maximum branching, it is about 200 nanometer. So you can imagine PVA which is a linear polymer, it shows a size of about 1000 nanometer. Whereas when you are producing branches using this PVA as a monomer, the size is drastically reduced, and particularly for V3, where you can develop about, if you can develop about say 70% of branching, then you can reduce that PVA size of 1000 nanometer to a size about 20 nanometer. So you can see that so much, I mean, variation you can do in the molecular size while you go for branching. So next slide, please. So again, the rheological characterizations we have done. One characterization is the flow behavior, mostly that creep characterization, that is shear strain with respect to time. And we can see for V1, the amount of strain is maximum, and it is giving a consistent strain behavior with time. V2 and V3, they also have, sorry, V3 have given maximum strain and consistent strain behavior with time. And V1 and V2 also, they gave consistent strain behavior with time, but the strain values are less as compared to V3. And we have done this swelling study also, and we have seen that the V3 is giving the maximum rate of swelling, and also amount of swelling is also maximum, and that is because of the voids that we have talked about. In case of these branch polymers, you can have so much of intramolecular voids. So because of that voids, we have recorded maximum swelling with this polymer. So this swelling experiment, this also gives us another structural implication of this sort of branched polymer as compared to this linear polymer. PVA is water soluble, it is linear, it is highly entangled and hydrogen bonded. So because of sorry, this hydrogen bonding, it is linear, and it is water soluble also. But in spite of that, it shows a lower, in fact, lowest amount of swelling because of its linear architecture and lack of your intramolecular voids. So next slide, please. Now, we studied the adhesion and growth of two kinds of cells. One is the your BMM, and another is the MSC. BMM, that is bone marrow macrophage, and MSC is that mesenchymal stem cells. Now, these why have we selected these two cells? BMM, that represented the differentiated primary cells, they are basically non-adherent. So adhering a BMM on a matrix or a scaffold is actually a challenge for their your growth adhesion, for their growth study. So that way, to adhere this highly mobile BMM cell was a challenge. And these MSC, these cell, these are stem cells and represented undifferentiated primary cells. Now, these MSCs, they required for making and repairing various cartilages and bone tissues, but these cells are much less migratory, although these are much less migratory as compared to BMM. Uh, but they required some special condition to satisfy for their adhesion and growth. So although MSCs, they are less migratory, but they are selective, highly selective. So for their growth study regarding the matrix. So that way we selected these two odd cells for their growth on this hyperbranch matrix. And next slide, please. And these are the results. You can see that it was evident that all the grades of these hyperbranch structures, mean V1, V2, V3, all, in fact, uh, they promoted the adhesion and growth of these BMM and MSCs. So that was a very important finding we got using this hyperbranched matrix. Whereas for the linear polymer, linear polymer, we did not find much growth. We have not included the study here. We have just shown for the hyperbranch grades for BMM and MSC, but for linear polymers, we did not find the growth of these cells because they were not adhering. So that it was an important observation, I would say. And the reason behind is that architecture, special architecture, branched architecture, which can accommodate these cells and also helps to supply the nutrients because of the your intramolecular voids for the growth of these cells. So that way, we could say that these branched scaffolds which can have a proper balance between the strength and your branching, they could act as a good scaffold for these adhesion and growth of all kind of cells for the regenerative medicine study. Next, please. So this is the last work that I'm going to show you, where we have developed polymersome kind of hyperbranched copolymers starting from ethylene diamine and trimesic acid, and we use that as a host for delivering alpha tocopherol succinate as a drug, and also we designed a drug transport modeling. So here we have used ethylene diamine and trimesic acid. The trimesic acid, it acts as that nodal point for branching, and these two monomers we have selected because they have a strong biocompatibility. Although we have experimented in vitro mode, but for the its potential application as a drug carrying vehicle and the release of the drug in vivo mode, for that potential application, we selected these two monomers for preparation of this branched architecture, branched copolymer. And most importantly, what we have observed actually, that these sort of polymers, they give a polymersome kind of structure. Now, polymersomes are basically a hollow spheres. They are having a thick outer shell and a hollow inside part, and these are mostly made of block copolymers, linear block copolymers, having a balance between hydrophilic and hydrophobic blocks. But here we were able to develop that kind of a polymersome like structure, and that was very useful for your as a vehicle for carrying the drug molecules and encapsulation and the your carrying, you're containing the drug molecules. So next slide, please. So again, we adopted this condensation route, just simply mixing trimesic acid with ethylene diamine in different molar proportions in order to have different extent of branching. And in presence of this acid catalyst HCl, in fact all the condensation polymers we made in presence of acid catalyst in order to have that accelerating effect in polymerization. And we carried out the reaction for 6 hours just by stirring and maintaining the temperature, and we formed the three polymers P1, P2, P3, and we loaded the 2% of TOS the solution with these polymers for encapsulation of this drug molecule inside it. So P1, P2, and P3, these are the three polymers we developed. And in P3 we have more ethylene diamine, and in P1 we have trimesic acid more to have more branching. So next slide, please. So this is the characterization, structural characterization, and what we have seen that as we have used more trimesic acid in P3, so we got maximum amount of branching effect in it, and from NMR we determined that branching extent to be 65% in P3, maximum branching extent, and P1 it was about 35%. We have not shown the P1 data. So P1 least branching with 35% branching, and P3 with 65% branching. So next slide, please. So here we have done this your rheological study. Rheological study is essential to characterize the hyperbranch structure and branched architecture. And we have seen that the more Newtonian behavior with this your branched architecture and maximum, we got it in case of P3 because of the maximum branching effect and minimum in case of P1. Particularly if you see the complex viscosity plot with the help of angular frequency, more Newtonian behavior would give you less effect of angular frequency on viscosity, and that just what we see in case of P3, and P1 and P2 showing a more effect because of the lesser amount of branching. Next slide, please. Yeah, this is the your DLS size, means the size determined from DLS, and we see that this P3, it is showing the minimum size, and P1 showing the maximum size, and these sizes are also we measured after drag loading, right? And post drag loading, we see an increase in size of these particles from the lower dimension to higher dimension, and that is because of the encapsulation of the drug. And we see the this TM picture, and from the TM picture you can see that the two layers of these spheres, the core is much dense and the periphery is much lighter, right? So this the two phasic structure like polymerism, as well as I was mentioning. In conventional polymerism we have a hollow core. Here we have a solid core, that is the dense core from where the branching starts, and the periphery where the branching ends is lighter. So here we have a polymerism kind of structure, but the reverse, with the reverse characteristics, that is the your solid core with light periphery as compared to the linear block copolymers that develops the conventional polymerisms. So after this drag loading, what we see that your there is I mean expansion in size of these particles, and while the drag goes out, there is a contraction and squeezing of the size uh, while the drag was released from these particles, right? So next slide. So here we have measured the drug release kinetics at different pH, and also from different matrix P1, P2, P3, where we have the extent or variation in branching. So we have different architecture, I mean the extent of branching as well as pH, the two variables to deal with to study the control release of this model drug from these polymerism kinds of nanopolymer particles. So go to the next slide. With these two variables, we have tried to develop a model for this drug release. Next slide, please. We have tried to develop this drug release model, whereby we have your used these diffusion phenomena as a controlling phenomena, and Fickian diffusion for modeling the drug. And next slide. Next slide, please. Yeah. So using this diffusion or transport concept, we developed this model. There was a calculation, and I have evaded the calculation from here. I am giving you the final model, that is dm kph plus into dB plus d. Now dB is the degree of branching, d is the diffusivity, and dm is the resultant diffusivity that we are getting from the matrix. And from this we have fitted the actual experimental data with this developed model, and we see that at different pH, we studied at pH 3 and pH 7, right? that is at your above and below the pKa value of the acids, where we have a difference in structure, right? To imbibe the pH effect and how it impacts the release. So what we found that our model was very good fitting. It was fitting very well with the experimental data that we have got for the different system, this P1, P2, and P3. And we also compared, next slide, please. We also compared with the conventional release models, that is Cosmere Peppers, Iguchi, with our experimental data, and we see these R square values is much lower. For Cosmere Peppers, it is around 0.88. For these matrices P1, P2, P3 at say pH 7, and at pH 3 also it is not very great, 0.75, 0.65, 0.8, like this. And Higuchi model also at different pH, the values are not too high, whereas the our model, we have this R square value at around 0.97, in the range of 0.97, 0.94, 0.92, means at least greater than 0.9. So that gives you an idea that our model was perfect in your describing the release kinetics of this sort of your nanopolymers that has the effect of pH and branching on the structure. So as I was mentioning that whatever this sort of drug release or any sort of these effects of your nanoparticle, nanostructure, and all that are being predominantly done with the linear polymers and your polymersomes. So those release kinetics and all, they will be effective, or they are in fact effective with these models are effective in describing the release kinetics from those conventional nanoparticles and all. But with these architectural polymers, these models will not work, and we have to design a separate model, an indigenous model, and we did it for this work. And in fact, let me tell you, we have developed other hyperbranched models with other drugs, and we tried to validate this model, and this model came very good for those systems as well. And we are very satisfied with this effort of us in developing a model from this kind of specialized architecture polymers. Now, once we confirm the, good functionality, good functional effect of these polymers in encapsulating the drug and its control release, then we tried to find out the characteristics of these and biocompatibility of this sort of polymers, and we did some tests for that. So next slide, please. So we did the cytotoxicity analysis with L929 cells, and we got very good values for this kind of test, and our polymers were very good. For these in not indulging any cytotoxic effects on these cells, fibroblast cells. So next slide, please. Yeah, live dead assay. So it also gave a good result that the your our polymers were good amount of compatibility. It has shown good amount of compatibility with these cells, and you see the bright green colors, which are the live cells, and you can see that the cells are not the cells are not died or with the application of this polymer. So that way we found our polymers to be very good for this sort of in vivo applications. Next. Yeah, rhodamine DAPI assay. It also gives negligible toxicity towards the fibroblast cells, and so that way it can be a good candidate for in vivo study. We have not carried out the in vivo study. We have planned to extend this for in vivo study. to see that what effects it gives regarding the cytotoxicity and your drug release effect. So here I would end my talk and finally I would conclude. Next slide, please. So what we see that shifting to branched polymers actually can facilitate the synthesis of both the nanopolymers as well as the nanocomposites. And intramolecular voids and chain-end functionality plays a key role in improving the functional properties actually. And since chain entanglement is largely avoided in the set of branched polymers, so such polymers and composites are not suitable for structural applications. So you have to take it for these sort of non-structural applications. And let me tell you that it would come in a very big way in these biomedicals and also in construction industry. Construction industry is highly benefited with the induction of these branched copolymers, particularly these superplasticizers. Because you see that these high rises now are being built in within a month, right? So that is because of the use of the superplasticizers, which has these branched architecture. So these are the two fields to watch out. One is the biomedical, another one is the construction industry, where you can see this effect of these nanopolymers and nanocomposites in future that are made from these architectural polymers. So here I would like to finish, and once again I would like to thank Dr. Jutika for this invitation, and also to all the participants. Thank you very much for your patience hearing. I understand that there were a lot of problems, maybe in communication and presentation, maybe because of this restriction nowadays we are facing in online mode. So instead of that, I hope you have enjoyed the lecture and the results that I have shown. And I hope this is kind of some sort of new information you have got regarding the architectural polymers and its possible applications, particularly in the biomedical field. So thank you very much to all of you.
Thank you, sir. Thank you very much for delivering the keynote lectures. Am I audible?
Yeah, I can hear you.
Yes, sir. So, and the topic is emerging. So, I hope the participants has got various types of new materials to do research and for empowering their knowledges. So, if anyone has anything or any questions to ask to sir, now the session is open for interaction. You can raise your hand if you want to interact with sir. Abdul Aziz.
Yeah, thank you, ma'am. Good afternoon, sir. Abhijit, sir. It was a nice presentation. We have
Good
got,
afternoon.
yeah, so it was very useful, sir. I just have one doubt. I was working on the drug delivery. I'm Dr. Aziz from Banarayam Institute of Technology, Tamil Nadu.
Okay.
I was
Okay.
actually working on drug delivery project.
Mhm.
While working on it, I was also doing this Higujis and Cosmeyer-Pepper's equation and all those things
Mhm.
with various pH.
Mhm.
One of the pharmaceutical people, they told me that this drug delivery study has to be done like first we have to check the delivery in salivary condition, that is
Yes.
pH of
Yes.
saliva, and then at the pH of the esophagus, and then at the gut,
Yes.
so
Yes.
intestinal.
Yes.
So should we do that, sir, or has it been done? Because I don't find much papers have that has worked on that way.
You
Uh,
actually see means this is what we do is that in vitro study, and you change the pH of the sink. I have not explained this experiment in detail. You are aware of that the sink pH you can check and maintain that with the help of a buffer, and you see the release study. So that will be
Yes, yes.
sufficient. So you don't require any paper to refer. You can just apply the pH of those organs and you can check the pH. So that is sufficient to get an idea about the release at different pH, that is at different organs.
Yeah, yeah. Okay, sir. Actually, I actually did like that only. But what they say is, I have to collect the supernatant of the pH, supernatant of the solution from saliva pH, and then I have to extract the drugs released in that. Because
Mm-hmm.
when we take orally, first the
Mm-hmm.
drug undergoes the salivary condition, and then undergoes the
Yes, yes,
esophagus, and then the gut. So it's
yes.
like they want it to be performed serially. So that is why
Mm-hmm.
I asked you, sir. Anyway, I will also try, and I wish to have contact with you, sir. Maybe I will
No
get your
problem.
mail
Anytime,
from the
anytime.
organizers. Yeah.
And I'm interested to know whether you have got good data fitting applying these Higuchi and Cosmere papers at various pH.
Yes, sir. Actually, I worked on that is
Linear
at
polymers,
pH.
right? You have used linear polymers.
I worked on nano emulsion, nanoparticle, and liposomes. I compared for the three
Yes, but
moieties.
all these are linear. So all these are probably
Linears.
your these are all
Mm,
linear. So for linear
yes.
polymers, these drug models will work. But when
Yes.
you go
Yes.
for these branch polymers, you will see
Mm,
that there is a huge deviation.
yes. Okay. Anyway, so thank you, sir. Thanks. That answered my question. Thank you.
Welcome, welcome.
Thank you, Dr. Abdul Aziz, for this question. Next question is from Arunagiri. I have requested you to unmute yourself.
Hello. Sir, good afternoon, sir. So it's an informative session. I have a doubt regarding whether this hyper branch polymer could be used for 3D printing applications.
No, 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 no. No, you cannot use it. As I
Oh,
mentioned, for 3D printing, you see, till now, ABS, polycarbonate, some polypropylene, these are familiar for this 3D printing because you need to develop high modulus, right? Because you will be printing in very thin mode, right? So you require very higher amount of modulus in order to have a very stable printed profile. So these polymers I mentioned, no, means these are devoid of your chain entanglement. So once you have that chain entanglement very less, then the strength of the polymer becomes very poor. So what actually we want to exploit from this polymer is their chain end functionality voids. So wherever these properties becomes useful, you have to use it, but not at least where you require higher strength.
Thank you very much, sir. I have no other doubts from any participant is there. Thank you very much, sir. Thank you very much for encouraging all of us with this emerging topic, because these polymer structures and all these are quite new area of research, and you have enlightened the various applications area also. So we all are empowered by it, and thank you very much for giving your valuable time, sir.
Thank you, and thank you once again for this invitation.
Yes, sir. You can left the meeting, sir, if you want, sir.
Yes, yes, yes. Yeah. I'm
Thank you, sir.
leaving. Okay, thank you, thank you.
Yes, sir. Dear participants, we have lunch break from 1:45 to 2:30. At 2:30, we have lectures by Dr. Hare Krishna Deka of CIPET CSTS-GUWAHATI. He will focus on different polymeric materials and their property along with the testings. So please join at 2:25. So now lunch time is there. So from 1:45 to 2:30, lunch break is there. And please, you can be in the same line or you can rejoin. Our link will be open. So we will meet again at 2:25 p.m. Thank you. Yes, attendance will be, we are monitoring the attendance by the software. So no any other seat will be there. So we are taking the attendance by us. So no need to worry for that. Just join the session. Welcome, sir.
Yeah, am I audible?
Yes, sir.
Yeah.
Yes, sir.
So I can start the presentation now.
Thank you very much, sir. And now I am going to give a brief introduction of Dr. Hare Krishna Deka, sir. He is the head of the Department of Training and Testing Department of CIPET CSTS-GUWAHATI. He is an expert consultant for different government organization and department all over the India in the domain of polymer. He is a gold medalist in polymer science from Tezpur University. He did his postdoctoral from University of Guelph. He has expertise on polymer composites, reinforced green polymer composite, bio-based hyperbranch nanocomposites for different antimicrobial activity and electrolyte activities. Sir has vast expertise on characterizing techniques of polymer material. Sir has worked extensively on national fiber and thermoset composites. We welcome, sir. We welcome you and thank you for delivering the session. Thank you, sir.
Yeah, thank you, Dr. Zutika Devi, for your nice introduction. So I am going to be. I'll talk today on the thermoplastic materials which are intended to be used for biomedical application. First of all, these all lecture will be in three category. First one, little bit about polymer plastics, then related to the biomedical applications, what are the criteria to be fulfilled by plastics material, and a general processing, how we produce a plastic material. So the third part I will cover for plastic processing technologies. So in these three category, I will explain for this one and half hour talk. So Dr. Devi, is it my slide is visible?
Yes, sir.
Yes.
It is visible, sir.
Okay, thank you. So if we go back to the different ages of human civilization, then we can see that the different materials, different ages are defined as the materials that we used in different activities, highly we used. For example, in Stone Ages, peoples are making different equipment, instrument with the help of these stones. Then with the growth of the human civilizations, we have discovered many different type of metals, and we did best use of those materials like bronze, then iron, then ceramic, different type of these ages we can categorize with the name of the different metals. And if we see today, from different applications, from simple to high-end applications, we are using different polymers. So it is my view that we can tell that this age is polymeric age. And to entrust my point, we can, if we see our daily life, starting from morning when we took the toothbrush, we use different kind of polymeric materials. That toothbrush is, again, we are using cloths, is where polymer polyesters are highly used. Again, in different automobile sector, entertainment, electronics, even sports, and different sectors, we are using different kind of polymeric materials. And if this can be supported with the per capita consumption of plastics, that in advanced countries they are using more than 100 kg, more than 50 kg. In Europe it is more than 65 kg. In USA it is 109 kg of plastic material, plastic products are being used for 1 year by person. And in comparison to those countries, if we see like your India, we are using around 10 kg of plastic materials per year per persons. And if we and the industrial growth of the plastic industries, especially these polymeric industries, are very high, and in some literature I have seen around 2.3% of expansion is going on in India in the field of polymeric materials manufacturing sectors. This is a special scenario for India, where the 30% of polymeric materials are composed of is coming from the sector of flexible and rigid packaging, including oven, check, and multi-filaments. In agriculture, we are using around 23% of polymeric products. In electrical and electronic application, it is around 14%. In household footwear and furnitures, we are using 9%. Building construction, 6%. Telecommunication and information technology, around 6%. In automotive, around 2%. This whole scenario is for Indian scenario, considering the Indian plastic manufacturers, industries, and companies. Now, you may be knowing the news that toy and sports sections, Government of India is giving a lot of interest to, specially to to produce the toys, so that the imported toys can be reduced. The number of imported toys can be reduced. So this is one of the avenue where within 1 or 2 years this figure is to go up. So this is the scope of different polymeric materials productions, where how the different industries, how it is growing in this sector. And if we compare the traditional things, this is the polymers which are gradually replacing those materials. For example, earlier we used a different kind of glass and metal bottles, or packaging different edible products like milk or oils, and which is now being replaced with 5-layer or different tetra layers polyfilm pouches. Again, oven sack bags, PP and HDP oven sack bags has replaced other earlier used like jute made cement or fertilizers bag. Even different laminates, which are earlier we used for with different kind of metals like aluminum or other materials, is being replaced with by different polymeric materials. So let us give me the definition in a very way. In a quick way, I also want to give the definition. The polymer, what is polymer? Polymer is nothing but a macromolecule which are constitute of organic, mainly it is organic or inorganic constituent, which are bonded together through covalent bonding, and that identical repeating units which produce the polymers are called monomers, and these monomers are covalently bonded together to make these giant molecules. And so the word like polymers, it came from Greek word poly and meros. Poly means many and meros means parts. The process through which the polymer is formed is called polymerization process, and the number of monomer repeating unit that monomer join together, that number is called degree of polymerization. And therefore, if we calculate this number of repeating unit with degree of polymerization, then we got the molecular weight of the polymer. Now, why this molecular weight of the polymer is very important? Because the molecular weight defines the characteristics, the properties of a specific polymer. The same polymer may be of same chemical linkages, all are similar in view of like chemical linkages, but the molecular weight, different molecular weight, same polymer will give the different properties of the same polymers. So we can also visualize like like the cooked noodles, which are a very long structure. with some coiling, which in term we call like entangled structures, which in a molecular level, and some polymeric chains are there which are linked with each other polymeric chains, which we call cross-link polymers. The properties of the polymers are different from the fine chemicals. Let us take one examples like solution characteristics. The other fine chemicals are easily soluble in solute with agitation and if necessary with some temperatures, but the same thing is not happen in case of, you cannot find in case of polymers. The polymer dissolves in a three-phase wise. First it will swell, then it will dissolve, then it will go for solution. It will make a solution. So in a three-phase manner, it makes a solution which is very highly viscous. So during synthesis of a polymer, we can also identify with the increase of viscosity of that particular solution. The comparison between the initial state and the final state will give you an idea about the formation of a polymer. In the definition, as we said, like a macromolecule or giant molecule, this is in some chemicals, like say for examples, ice cube. Due to the hydrogen bonding, the structure of the ice cube also becomes very big. But this is just a physical characteristics, not the chemical inherent properties. Therefore, if you heat it, the shape, the volume will decrease, and even if you go for more higher heat, then it may again evaporate. The same thing is if we compare with the polymers, you can find that the inherent chemical linkages itself it makes it a giant molecules. So this is one of the major differences. In that way, we can find different properties which are peculiarities which makes a very clear differences between a fine chemical and a polymers. Some of the examples just I want to show, like vinyl monomers, phenylidene monomers. Vinyl monomers where the structure is CH2 double bond CH2X, and if we replace this X with hydrogen, then it will become polyethylene, where we use the symbol PE. If his X is replaced with CH3, it will become polypropylene. From this ethylene or propylene monomer, we can get polypropylene. And in that polymeric structure, if we replace this X with Cl, chlorine, then we'll get polyvinyl chloride. If we replace with a phenyl group, it will be, the polymeric structure will become polystyrene, etc. And let me also give you another monomeric structure, phenylidene monomers, where X and Y, if we replace with say CH3, then it will become polyisobutylene. With chlorine and chlorine group, then polyvinylidene chloride. This is all are the polymeric structure from monomer to polymer. And if X is replaced by phenyl and Y is replaced by CH3, then polyalpha methylene styrene. Another very important polymer is polymethyl methacrylate, where X is replaced by CH3 and Y is replaced by COOCH3, polymethyl methacrylate, etc. So this is just to give you an idea how from the different monomeric structure we can get the polymeric structures. Now, if we see the classification of polymers, based on the criteria, we can divide this polymer in different categories. I'll mention only few. Say for example, based on the physical properties, we can divide the polymers into three categories: rubber, fiber, rubber, and plastics. Where fiber is having very high tensile strength, L by D ratio is very high. In case of rubber is having elastomeric properties. Its tensile properties are less than fibers, but it's less than fiber and plastics. In the plastics, which is having viscoelastic properties, where its strength, if we consider the strength, its strength is higher than rubber, but it is less than fibers. Again, based on the source, we can divide the polymers into three categories: natural, synthetic, and semi-synthetic polymers. The natural polymers, the source is we derive the polymer from natural. Synthetic means it is a lab-based polymers, lab synthetic polymers, and semi-synthetic are the naturally obtained polymers are modified, so it is called semi-synthetic polymers. I have given a very big table where it is shown that depending on a factor, we can divide the polymers. So depending on properties, depending on source, depending on again thermal responses, we can divide the polymer in two groups: thermoset and thermoplastic. So similarly, the depending on formation mode, we can divide these polymers in like three categories. Basically in three categories: addition polymers, condensation polymers, rearrangement polymers. Let me show you the formation techniques. Like addition polymers, where the monomers are combined together with through covalent bondings, where the main criteria for these addition polymers are the retention or the chemical linkages are similar as that of the monomers. If we see the condensation polymers are the polymers where after formation of where they go for a chemical reactions, and after formation they lose a very low molecular weight molecules groups. Here dialcohol was reacted with dicarboxylic acid, and they have formed a ester linkages, and at the same time they have eliminated one water molecule with each of the reaction. Now the rearrangement polymerization techniques are those techniques where chemical groups molecules is rearranged itself, and at the same time they go for a chemical reactions. Here the polyol and diisocyanate, the example it was shown here is like polyol and diisocyanate, where you can find out the OH is reacted with this isocyanate group, and at the same time this hydrogen from the polyol is rearranged itself, and after rearrangement it has linked with this nitrogen of diisocyanate, so NHCOO. This polyurethane formation is a rearrangement polymeric reactions. As I have mentioned, the different applications, the sector-wise applications. Just I want to show few of the examples in 2 or 3 field application sector. Let me first give you the application of polymers in agricultural field. In agriculture, starting from greenhouse covers, then mulch film, then the different bell wraps, then the different type of bell net covers. Nowadays we are using lot of drip irrigation agricultural system, where irrigation starting from the pipe that which carry the water till the drip tap. We are using different type of polymeric products. In automotive industries, like interior parts, for example, different mat and cover, different knobs, then airbag covers, different than your grommets, and again under the hood we are using different type of polymeric materials. So interior, exterior, under the chassis, inside the bonnet, we are using thousands and thousands of different type of plastic polymeric materials, polymeric products. Biomedical sector, I'll go through, I'll show you a different table where we are using biomedical, in biomedical applications, where what are the different type of polymers we used. A variety of polymers are used in medical sectors as a biomaterial. It starts from, say, like a facial prosthetics to tracheal tubes. Again, in higher applications, we are also using kidney and liver parts to different heart components. We are also using for denture applications to hip and knee joints, different medical sealants, different medical adhesives. Here, a few examples was shown, like polyethylene, which are used for joint replacement, polyethylene terephthalate for vascular prosthesis application we used, polytetrafluoroethylene PTFE we use for the vascular prosthesis as well as soft tissue augmentations. I want to mention that PVC we are using for tubing as well as different type of blood store vessels we are producing from PVC, as well as from polyurethane also used for different tubing and blood containing devices we used. Polymethyl acrylate are highly used for as a cementing joint replacement, as a cementing material, as a also used for dental restorations. Silicons, different hydrogels, these type of polymers are used for different biomedical applications. Now I'll discuss on how we select plastic materials for biomedical applications. What are the different criterias with respect to different conditions? Basically, when we choose a polymeric or plastic material, the three things we have to see, like first, the chemical linkages or polymeric structures. Number two, its property. There are different required properties in terms of physical or chemicals. Number three, the environment where this polymer will going to be exposed. Again, so in these three categories, first we need to need a checklist. Let me go through one by one. Let me consider first the environmental exposure conditions. There are a few checklist it is given. Number one, should the material be biocompatible? Because if the material directly exposed to human body, then it need to be go for biomedical test, biocompatibility test. Second, will the component be in contact with the body tissues or drug? If so, then for how long? If the first answer is yes, then we'll come to the second question. Then if it exposes to a body tissue or to a drug, then again we need to know for how long. We generally categorize for three categories. Number one, below 24 hours, then above 24 to 30 days, and the third category is above 30 days. So that thing we have to know. Third, is the product a single-use instrument or device? The product may sometimes it may happen that the material will be used for instrument or devices. Then in that case, again, we need to know where that material, that product will be used for instrument or it go for exposure of the human body. Then will the component undergo sterilization? If so, which method of sterilization will be used and how frequently will it be sterilized? Because different polymeric materials is having different chemical resistance. Their chemical resistance is different for different products. Therefore, the sterilization techniques is also important, and The frequency is also very important. Then will any other component, chemicals or solvent, vapor be in contact with the product as a part of hospital cleaning? Because in hospital cleaning, we are using different cleaning agent that may again hamper, or it may again deteriorate, or the resistance power is a very important factor to be considered here. Then will the device be painted and electroplated or glued? Because paint means it is also a different kind of chemical. Electroplated means it is a different kind of process, and glued means also we are using different type of techniques. So this thing had to be again need to be clear. Then humidity and temperature also plays a very important role for the longevity of, to consider the longevity of a product. So the environmental conditions, like especially for humidity and temperature, plays also an important role. Then the dimension stability in a wet or humid environment is also critical. If we are using the material in a humid conditions, then how long it will retain its the dimension is also very important. They must have the tolerance. It must meet the tolerance with that specific environmental conditions. Then in some of the devices or equipment, we are also using different type of rays, X-ray or UV rays. Therefore, the product which are used for different fabricating different machineries for UV light or X-ray light or different kind of fluoroscope instrument. So the plastic parts may have the power to resist those high frequency energies. Is that so in some of the equipment, again, other properties like, say for example, outdoor exposure, exterior part, or it may need some special properties like flame retardance, that colorability. These all factors again we need to consider. Then second characteristics. So these are all like the different checklists that we need to clear to consider for selection of this plastic polymeric material for to be selected for biomedical sectors. So the second thing, again, functional and mechanical considerations. The check points are, what are the overall parts dimensions in the sense like dia, length, or with whatever the dimensions it will be there. And is the dimension stability a factor to be considered? Sometimes it may also happen that the dimension may not be a crucial point. But if it is yes, then again we have to go for another points that need to be clarified. What are those? Those are like, what is the maximum stress on that specific part? What kind of stress we are applying to that part? It may be tensile stress, it may be flexural. So depending on the again applications, whether it is a load bearing capacity, where we are pulling, we are giving a stress, flexural stress. So depending on the applications, again it had to be clarified. If the first question is yes, answer is yes. Then other mechanical related properties, for example, toughness, impact resistance, that has to be verified, and the depending on the applications, that these properties like toughness and impact resistance may again play a crucial role. Again, if that application demands electrical insulators, then again from the property point of view, again that electrical insulation capacity need to be checked. Again, we like depending on different manufacturing process, the final product is also differ sometimes. For example, the injection molded component, we can make by micro molding or insert molding, or we may also go like gas assisted molding processes. So the different manufacturing process also play a role of the final properties of the products. And overall, if we consider all these factors like different properties, mechanical property, like say tensile strength, elongation properties, toughness properties, again impact properties, then after summarizing, again we need to see what will be the cost of the final component, because this is also one of the major important, that it must come within a specific cost estimation. So design part, the projected life with the part, and the design is also related to this cost. Some of the while choosing the polymer, if the polymer need to be a biocompatible, if it exposed to exposed to our body systems, then it must go through the biocompatibility test. And for that purpose, especially the different test standards are there. USB class 6 and ISO 10993. In most of the cases, in general, the ISO 10993 is highly used test conditions, and these test conditions specify the process that contaminants, residues, and degradation of the products while doing the biocompatibility test. It's a very stringent and it is one of the very suitable test for biomedical devices. As per the ISO 10993 part 1, for selection of biomedical products, a summary sheet is shown here. In the extreme right left hand side, the 3 category is given. The medical products which contact to the body surfaces, number 1. Number 2, medical products coming into the contact with the interior of the body from outside. And number 3, the products which are implantable to a body system, implanted to a body system. And for these 3 different applications, categories applications, how it come into the contact of our body, whether it will come, if it is yes, then how it will come to our contact body. So depending on that, we are categorizing it in three category. Now let me show you example wise. Here A, B, C, as I mentioned earlier, like A means like a short-term duration, will less than or equal to 24 hours exposure time. B means less than 24 hours, sorry, greater than 24 hours to 30 days. And C means more than 30 days. So any product which is coming into the contact of skin and which is to be exposed for a short term, that means like a maximum of 24 hours, then we have to do 3 tests. Number 1, cytotoxicity. Number 2, sensitization. And number 3, irritation test. Again, if say for example, if it goes for an exposure of more than 24 hours, then additional test we have to do, like acute toxicity, subchloric toxicity, and genotoxicity, and implantation test. These 4 extra tests again we have to carry out. So here in this table, again, the product, the polymeric product which will come into the contact, say for example, if it is coming into the contact of a blood system for a minimum of 24 hours, for a maximum of 24 hours, then again these 4 tests we have to carry out: cytotoxicity, sensitization, irritation, as well as acute toxicity we have to carry out. For products which are implanted in a body system, for example, say for tissue culture, for bone replacement, if the product is going to an exposure of less than 30 days, then these all 7 tests up to this implantation test need to be carried out. So this table summarizes how, for selecting a product, polymeric product, depending on their exposure time, depending on the way how they expose, that the different properties need to be carried out. So this is a very clear table that it gives a very clear ideas. Now, after summarizing those this table, some of the application wise, the different polymer what is selected is given here. For non-contact with human body, which do not go for any contact of human body, say for examples, different syringes, blood storage bags, glucose drip bags. For that kind of application, the polymer selected which are found suitable are PVC, polyamide, PE, polyethylene, polystyrene, and epoxy resins. For short-term contact with human body, for example, the different products are which are catheters, feeding tubes, drainage tubes, surgical instruments. For those type of applications, the suitable polymers are silicon rubber, natural rubber, PVC, polyurethane, polyester, polyether ketone, polyphenol sulfone, nylon, Teflon, etc. Again, for medium term contact with human body, for example, cultures. So in that type of application, polyester or PP or nylon are suitable. And for long-term contact with human body, for example, drug delivery devices or say for any plants, the suitable polymers are polyurethane, silicon rubber, PMMA, different hydrogels, polysulfones, polydimethyl siloxanes, etc. So after these polymers are found suitable with these different according to the different exposure and time of exposure of the human body. While considering the selection of the plastic material, this is also very important to see how the drug flow, in which direction the drug flows. When the material is in the direct contact with the flow of the drug through any device or through any device, it also plays a role. And chemical resistance, as I already mentioned, it's also very important that what kind of drug we are using, because it may again started the polymeric material degradations. And another important requirement for most of the medical devices is the sterilization compatibility, because in sterilizations we use water, we may use different chemicals such as ETO, we may use different kind of radiations, we may go for autoclave. So the polymeric product which have used in the medical devices must have the sterilization compatibility. If we are doing for a, say for example, if we are going for an autoclave, the minimum temperature, melting temperature must have more than 100 degrees centigrade. If we are using if we are sterilizing through radiation, the polymer must have high radiation resistance. If we are using with chemicals, it must have chemical resistance properties. So that compatibility is very, very important. From compatibility side, if we consider the sterilization compatibility, some of the polymers, say for example, polyether ketone, it shows very high chemical resistance, very high water resistance, very high radiation resistance properties. Again, for example, if we see like a polycarbonates, the high energy resistance is not very high. It's just an average resistance it shows. And again, if we consider the polysulfone shows very good high radiation, high energy radiation resistance power. It shows good to moderate, again, chemical resistance, and it shows high hot steam resistance and high hot air resistance capacities. So this that sterilization compatibility is also plays a very major role when this sterilization is one of the process that need to be carried out in a different frequencies. So this is what I already mentioned, like the environmental wise also, when we are using very different chemicals for disinfection chemicals, then it must have resistance to those chemicals. Mechanical property wise, depending on climate conditions, depending on where to be used, depending again during the transport also, it may go for a damage if it does not have proper impact resistance properties. Yeah, so depending on, so therefore for selection of this polymeric material is basically we divide in 3 groups, and just I want to give you an example of a polyester while considering all those factors. Polyesters, in the last portion of this table you can see like it is mentioned PGA, polylactic acid, polyhydroxybutyrate, PL, PCL, PPC, PBS, lot of different type of polyester it is given. Accordingly, their tensile properties, tensile strength, modulus and elongation is mentioned. And now if we want to use these polyesters for, say for example, tissue application for bone replacement, then the minimum strength modulus that is needed is 483 megapascal. The tensile strength starts from 2 megapascal, and elongation it's need 2.5%. Elongation at break it needs 2.5%. So while selecting that, while knowing this criteria, we can select the different type of polyesters for these tissue applications. For example, say we can start from PBS because it is more than 483. Its tensile strength is also, it is 17.5 approximately. Elongation and break is also 6%. So we can substitute depending on the nature, environmental nature, what other extra criteria needed. So depending on that, we can again replace those materials. So this is how we can select a material. First, we need to know the required condition, then we can evaluate the different properties as per the needed criteria. The degradation pattern is also very important because the lifetime of the product need to know before we use the polymers for the specific purpose. And for polyhydroxybutanoate, few of the polyesters that is shown, say for example PLA, polylactic acid, L or D both, which are degraded like 50% within 1 to 2 years. Then the next 12 months it goes for 98% degradation, and in other 12 months it goes for 100% degradations, and it degrades through hydrolysis process with the action of enzymes. Therefore any chances that leads to a hydrolysis process may affect the degradation pattern of the PLA. Another example, say for example polyhydroxybutanoate, its 35% degradation takes place after 6 months, and another degradation takes place 60% degradation takes place after 24 weeks. If the it also depends on the thickness of that material, but roughly it's 60% degradation takes place after 24 weeks, and the process is hydrolysis process. It is found from that literature that non-specific esterase enzymes through the non-esterase enzyme process, it goes for degradation. It shows the degradation behavior. So degradation behavior and the time of degradation is very important to know, because ultimately it will define the total lifetime of that products. And these all degradation pattern is in vivo degradation. This rate are all mentioned as in vivo degradation rate. Another biomaterial we want to discuss is polyurethane. So here a few of the things that in my research career we carried out, that also I want to explain. What is the urethane linkage? Urethane is not NHCOO. It is a very versatile material due to its chemical linkages. You can say like the urethane linkage is act as a hard segment, and the soft moiety is the carbon chains. Therefore, if the total material itself, it shows a variation of hard segment and soft segment, like a combination of hard and soft segment, and this hard and soft domains, it renders special characteristics. It make it as a versatile material, and it shows very high abrasion resistance, tough and impact resistance, having high tensile strength, tear resistance, excellent weathering and chemical resistance. The things that I was discussing over the previous slides, like resistance to chemical, resistance to weather, like moisture, like temperature and moisture at same time. It also shows high load bearing ability, adhesive strength, and the main important point is tailor-made behavior we can produce by choosing different soft moiety, by sensing this soft moiety, soft segment, by sensing the length of the soft segment, we can get a tailor-made properties from the polyurethane. In my research career, we have selected one oil which is locally available, which is Mesoephria linseed oil. It is having 70% of oil in comparison to the total weight of the seed. That product, again, we want to make is a hyperbranch polymer, because it ultimately our aim was to make it to nano composites, so that the nano things, the nano fillers can be trapped inside the hyperbranch moieties and that makes a very stable and high duration nanocomposites. So that was the motive, and that's why we have selected hyperbranch polymers. These hyperbranch polymers are three-dimensional, globular, non-entangled, highly branched structures. The advantage is it has large number of terminals active groups, and due to this globular structures, it shows lower melting and solution viscosity. And at the same time, due to the presence of the lot of terminal groups, its solubility characters is very high. Therefore, the processability, the handling is very easy. It becomes very easy for this type of hyperbranch polymers, and at the same time, we can choose this metric as a matrix for making different composite systems. So here, the monoglyceride component was prepared from the natural oil, and then it was blended with polycaprolactam with some specific percentage. This was acted as a blended hydroxy system, and this blended hydroxy system was reacted with diisocyanide. Then we find out then in the synthesis route was selected in such a way that a preform will be formed where the isocyanate, unreactive isocyanate groups will be there, and in a second step with the pre-polymer, a glycerol moiety was added. Glycerol is, we know, like glycerol is a industrial waste material. The purified glycerol was reacted with these unreacted pre-polymers, isocyanate pre-polymers. Then due to its presence of three hydroxy groups, we finally obtain a very highly branched structures, and the different type of surface active groups. Here it was shown as L linear group where one hydroxy group is present, then terminal where two hydroxy group is present, then a dendritic unit where no functional group is present, but it is highly with these three axis, it is with these three directions, all the active groups are joined together by forming isopolyurethane linkages. So these highly branched structures is produced after reaction is completed. The property shows the different mechanical properties, shows like tensile strength is more than 5, elongations are more than 400 degrees centigrade, hardness is more than 70 Sore hardness. It shows very flexible materials. It is the impact resistance test is also very good, and it also having high adhesion characteristics. At the same time, the thermal properties shows the degradation pattern where it is seen that more than 340 degrees centigrade up to 340 degrees centigrade, no degradation pattern is observed for this polyurethane. In the next step, this hyperbranch polyurethane was utilized to form a nanocomposites. Here, different kind of nanofiller was used with a motive to be used for biomedical applications. First, silver nanocomposites was prepared. Here, the beauty of the nanocomposite was no reducing agent was used during the synthesis process. Thus, the matrix systems is itself act as a reducer to convert this silver to the nanosilver stage to zero stage. And after formation of the nanocomposite, we have also studied the stability test. It shows very highly stable conditions. The peak in UV it was taken from different time period, which reflected the stability of the form nanocomposites. The biomedical application, for the biomedical applications, the cytocompatibility test was done with RBC hemolysis protection assay. These proved that the percentage of hemolysis inhibition is increasing with up to certain level with the incorporation of the incorporation of the silver. The antimicrobial activity is also very potential it shows the potential applications for antimicrobial activities, because the assessment shows the highly antimicrobial resistance power with the matrix systems. Therefore, the prepared silver nanocomposites is having highly potential for to be used as antimicrobial biocompatible applications. In the next step, we use this hyperbranch polyurethane matrix for the formation of iron-based nanocomposites. The target of making the formation of this hyperbranch iron-based nanocomposite was to use as free radical scavenging applications, and the iron-based nanocomposite, it shows 65% of scavenging capacity, and the 10 images also shows very small. It comes to like 10 to 20 nanometer length iron nanocomposites, which are distributed uniformly throughout the matrix. The mechanical properties is all the other 10. The mechanical behavior is a very important sector, and that mechanical behavior was also studied, and it shows that the maximum of 11.51 megapascal tensile strength it was achieved, and the maximum elongation around 730% we have achieved in these different nanocomposites. A potential applications for using potential application for using this hyperbranch polyurethane was to make a multi-wall carbon nanocomposite. Here the motive was to make a safe memory application. The safe, and the material we prepared a ring which was freezed in 0° centigrade, and an external factor, heating factor, was given again, and we find out the safe memory behavior is shown with by the hyperbranch polyurethane multi-wall carbon nanotube nanocomposites, and the retention, and then at the same time the biocompatibility test was also carried out with the RBC hemolysis assay at the cellular level, and it was found it is non-toxic. And further, the biodegradation behavior was also studied. Which was exposed with different bacteria. The same picture shows the growth of different bacteria over the surface, and it is degrading. You can see the different degraded surface. These all are degraded surface, and these all are bacteria. With time, the growth of the bacterial growth are increasing. That means it is deteriorating. It is degrading the surface of the polyurethane composite, and it is degraded the material. So the three different aspect: number one, safe memory; number two, biocompatibility. Number 3, degradation pattern was studied. Therefore, and the study shows that these polyurethane nanocomposites is highly have potential to be used for safe memory biocompatible biomaterial. The other properties like thermal properties was also studied, and it shows the improvement of course, the multiple carbon nanotube has very high thermal resistance, but with the incorporation of the multiple carbon nanotube, the initial degradation pattern shifted towards higher temperature in comparison to the hyperbans polyurethane matrix. Yes, one of the property that is very important was rheological behavior. This rheological behavior has revealed that the formed polymer composites shows a temperature dependence property. That means the processing, the behavior, with the existing conventional equipment, it can be fabricated. Now, when we have mentioned about the processing characteristics, once we fix the polymers, once it is selected for a specific applications, again, what kind of processing techniques we'll use, it is very important and we must know. And already the study is there, like say for example, for say for example for polyethylene. So just now I have mentioned about polyester and polyurethane. So say for polyurethane, the process like injection molding machines, like extrusion process, like rotational molding process, then foam molding process. This process it can be carried out with the help of this technique process. It can be the different products from polyurethane can be prepared. So selection of processing techniques is also very important. I have also mentioned in my above slide that depending on the type of processing techniques, the final properties also may vary for the same components. So processing technique also a very important thing. So while for making different processing techniques, the process is first we need to heat the raw material, plastic raw material. We need to heat it. It must go for a softening. It must soft it. Then second step is shaping or forming. Then under some constraints, we give them a shape, and then third step we cool it to get it to retain the given shape. So this process we follow. And for processing also, what kind of size, the size of the product, the quantity of the product, the rate of production, the required dimension accuracy, the required surface finish, the required form, and the nature of the raw material. So depending on that, again, we are using different type of processing techniques. What are the processing techniques in general we use? Number 1, injection molding process, extrusion techniques, blow molding process, compression molding, thermoforming, rotational molding. I'll cover these techniques one by one. What are the prerequisite conditions, or what kind of process we follow in these different type of processing techniques? So in molding process, the resources are 4M. First, M stands for material, selection of proper material, selection of proper machine, selection of proper mold, and then experienced manpower required. Now, while we'll be going through these techniques, one terminology will come: mold. What is mold? Mold is the part which gives the shape of the product. It has four main functions. It receives the material from the machine. It distributes the material throughout the shape. Then it gives a proper shape. It forms as proper shape. It has the provision for cooling, and finally it disposes the material. So this is the function of mold. I'm showing you one picture of a mold. That's this metallic part is called mold. This mold is having two main parts. One is called core plate, another called cavity plate. This core and cavity, when it joins together, then it forms a cavity inside this mold, where this plastic material is injected so that it will get a proper shape. Pictorial diagram, it is here. What are the different components of the mold? In a mold we have different component we call runner, gate, guide bush, guide pillar. This is the guide bush. This is the guide pillar. This is the gate through which this is the runner, where this material here it will enter, raw material, melted material will enter, and finally it will get the shape. This is a picture of a mold which are used for injection molding machines. So what is injection molding process? Injection molding is a manufacturing process where a hot melted polymer is injected to a mold cavity. I just few minutes back, I have shown you what is the mold cavity. So into a mold cavity with the shape of the piece that will be produced, the molded material is pushed to the cavity with very high pressure, and we allow it to cool down for some time, and then it is ejected from the mold. The injection molding process is therefore it is having these 6 steps. First, mold clamping, that injection. I'll show you one with a pictorial diagram. Injection, then hold on, then refilling, then mold opening, and then finally ejection. Let me give you let me give you more clarification on the process, the sequence, the machine operation sequence. First, the mold are closes, and this screw. I think I also have one video. I can show you the video so that it will be more understandable for you.
The following sequence illustrates the basic functions of an injection molding machine and the process by which plastic pellets are transformed into molded parts. Pellets are fed into the hopper. A screw conveys and melts the pellets within the barrel. Melted polymer is forced into the closed mold. The parts are formed. It's cooled within the mold. The mold is opened. It is ejected or removed from the mold. And the cycle repeats.
Yes, so yeah, so the first we close the mold, then the screw in these injection machines, it moves forwards, and it pushes the materials, it materials to the cavity. Here the cavity is filled up with the material, and then it puts the material so that the whole cavity will fill up. Then once this cavity is filled, then it comes slowly, it comes back, and at the same time the other raw materials is prepared for the next shot. Once the cavity is filled, the gate is freezes off, and the product which are filled in this cavity is given some time to cool down, so that the dimension, it retains the dimension stability. And finally the mold is open, and it is ejected from the mold, and then again the same process, it's again the same cycle it starts. Once this is ejected, the mold is closes, and again the screw, it starts filling up the material. That means thus it becomes ready for the next cycle. In some of the equipment we see, we find that it is having two different color or two different material is there. So it is fabricated through co-injection molding. The co-injection molding, it is a terminology, it is described for the process of injecting two chemically compatible polymer melt one after other into the same mold through injection molding process. And the material which comes first, it is called core, and the material which on the top of that, it's called skin material. The core and the core and the skin, it's injected through the nozzle. Here the picture, it shows the core material, it goes first, then the core material goes first, and then skin material goes on the top of the core material. Sometimes we can find some material, some product is having two different color or two different materials. The only condition is it must be chemically compatible. Another process is extrusion molding process. It is a continuous process. The plastic raw material is continuously plasticized by means of screw plasticization action, and the melted material is continuously pumped out from the machine through a standard die. Now, in case of injection molding machines, we used mold so that we can get the final product. Shape. In case of extrusion, here we use a standard die which gives the final shape of the product. Once we get the shape after the die, then we give sufficient time for cooling and sizing systems. The different products like film, pipe, tube, profile, monofilament products are made with these extrusion molding processes. Now, as in this picture, I just want to explain the action of a single screw extruder process. Here it is the hopper. Through hopper we push the raw material, and the screw continuously it rotate. Here different heating and cooling system are there. We use the heat for melt the materials. By the action of rotation, rotating the screw pushes the material towards the die, where the die defines the shape. This die defines the shape of the final products. An extrusion process by which we make blown film is shown here. It is having different components. First is extrusion process. where the raw material will be plasticized. Then this material will be pushing towards sizing units. Yeah. Here it will be pushing towards a sizing unit, and then after this die, some air will be given, which will blow the tube. It will make a tube-like structures, and then when we reach to the required thickness, wall thickness of the tube, we will just roll it up. We will roll it, and finally it will be wind up. It will wind up to get a complete roll, and as per the required length, we just cut it. For blown film extrusion, again two different techniques are there. One is up blown film, and second one is downward blown film. For mainly when we need some kind of clarity in the product, then we go for downward blown film. Downward blown film is the hollow structures. It will blow towards downside. This is a factory process where the extrusion process is somewhere here, and we after extrusion it will blow up, then it will go upward side, and it will wind through different rollers, and finally it will be rolled up in a final structure. A second process for extrusion is called cast or flat film process, flat film extrusion process. Here the melt which is coming out from the die will be will chill through a rollers, where it will go for solidifications. The extrusion portion here we have not shown. After once the extrusion process is coming out here, after the die, immediately it will be chrome plated seal roll. It will go over this chrome plated seal rolled. Here water or different coolant will be running continuously, so immediately it will be going for solidifications, and then we'll just trim out the necessary things, and it will be on the winders. This is one of the machinery, the picture is shown machinery which really make the cast film. Now another important is for making different tubes or pipe system, because I have given some of the examples like catheters or other which are having tubular structures. For making those tube, especially like when the wall thickness and flexibility, then different rigidity differences are there. So depending on the different structures, we go for pipe extrusion processes. In pipe extrusion processes, different components are here. It is shown in the pictures. It's a very rough picture that I have shown here. First, extruder. Here in this side, the extruder will be there. After the extrusion, where after the plasticization, the extruder, the material once it is come out from the die, then it will have to go through the sizing unit, where it will define the different size, different the MOD, outer diameter MOD, it will be defined here, and then once the sizing is completed, it will go through some cooling tank, where the the shape that was given in the sizing unit will retain, it will get fixed, and then depending on the length or depending on the nature of the pipes, depending on the length of the pipe, either it will be cut down or it will be roll out depending on the wall thickness, depending on the MOD, etc. Now again for sheet extrusion, processes, the melted polymer once it is coming down from the die, it is again going through the sizing unit, and the different process that, like say calendaring, cooling rolls, and then finally here you can see here in this picture, finally the sheet structure it will be given. Another, so first injection, second is extrusion, and another important point is now like blow molding techniques. The blow molding techniques, it is divided basically in 3 groups: extrusion blow molding, injection blow molding, and stress blow molding. I think I am also having one video on blow molding, I guess. Yeah, let me first show you this blow molding, then we'll go for this. Yeah. So just now we have seen that video, like how a blow molding process techniques is how we operate fabricate the different blow molding products. Here, depending on the process of blow molding, we divide the process in 3 groups: extrusion blow molding, injection blow molding, and stretch blow molding. The extrusion blow molding process, once we first, it will go for plasticization process through an extruder. After the extrusion process, it has to pass out through a nozzle, and it will form a required length Parisian. It is like a hollow structure, and hollow structure. Then this Parisian will be depending on the required length of the product, it will form, and then the mold will close around the Parisian. It will just close up in that video that was shown. So from the bottom of the Parisian will be sealed by firm closing of the mold. The mold will come from 2 sides and it will close up. The bottom will seal through by the action of mold closing, and from the top the compressed air will be given. The required the required pressure will be given, compressed air will be given, so that the Parisian will fit into the shape inside the mold that it was closed together, and then sufficient time will be given for cooling and it will be ejected, and then again it will continue the same process. After extrusion, molten materials will be coming out from the extrusion through the nozzle, and it will form the required like Parisian. the mold will clump together. It will close the mold and it will block the bottom side of the perisson. From the top, the compressed air will be given, and then it will be blown out so that it will stick to the wall of the perisson. Sorry, wall of the mold. Here the same process was given through this extruder, a tube-like structure which is called as perisson. It will come, then this mold will close together, but the bottom will be sealed. Then from the top, compressed air will be given unless it will stick to the was inside the wall of the closed mold. It will be given sufficient time, and then it will be ejected. The basic process for injection molding machines, for especially PET was highly used, and all the PET bottles which are used for different our day-to-day life activities as well as for biomedical application also is formed only by this technique. The video was also shown in that for PET preforms. Then second category, stretch blow molding. Stretch, let me go directly to this picture so that it will be easy for you. Stretch blow molding. First step is conditioning. We melt for this actually we need a preform. This preform is again it is prepared through injection molding process. This preform, once we get the preform, say for example here this blue color, it is called as preform. This blue color structure, it is a preform for making this shape for this making this bottle, specific bottle, specific size bottle. So this preform, first step we need to do a preheating, pre-required heating. We need to little bit soften so that once we give compressed air, it will blow. Once the preheated is over, we fix it through a core rod which is coming from the stretch blow molding. Then we close the mold. This is the mold. So once it is closed, then we give compressed air, and once we give the compressed air, this is the sequence wise what happens. It is shown. First, it will stretch from sidewise and lengthwise. It will start stretching. Here you can see stretch from wall side, from here wall side, and also from downward, and then it will finally stick to the shape of the mold. So that's why this process is called stretch blow molding. Here two action is simultaneously working. First is stretching, blowing, then is stretching. Nowadays what is happening is like injection stretch blow molding this technique is also highly used, where the injection and stretch blow molding was just combined together. Instead of using the preform in a separate technique, in the same process, first with the help of injection molding machines, the preform is fabricated. It is conditioned inside. In the second step, it is conditioned inside the machine. Then it will fit with the mold, with the core part. Then blow will start, stretching will happen, then it will get to the fixed shape, and then ejection. So this technique is called injection stretch molding machine. Here different, as I told you, like different preform photographs was given. From this preform itself we can get different shape bottles. And in the left hand side, in my left hand side, the mold which is used for making this preform was shown. technique is a technique called compression molding process, which is also a very important process. The compression molding process. In that process, the material we get it either in powder form or in pellet forms, and that pellet is heated up to a certain temperatures. Then it was given in a specific temperature and pressure combinations. It was the material is given a shape. In general, this compression molding process is used for thermoset materials, and like phenol formaldehyde or urea formaldehyde or melamine formaldehyde. These thermoset compounds are used for making different sheet-like structures, and given a very big structures. In generally, we use the compression molding process. Another technique is the vacuum forming techniques. In this process, the different steps are first clamping. We like here we first from in my right hand you can see, in my left hand you can see like the clamp. First we give the clamping here, and then the different preheating we need it. Then pre-stretching is required. Once we give the heat and stretch, then we seal it by using vacuum. And finally, the once we give the vacuum, the mold below the material it just stick on that material, and then we give the cooling time, sufficient cooling time, so that it will set on that product, and then finally we eject the material. So this is these are some of the processes, some of the processing techniques that commonly used for fabricating different plastic products. Basically, it is mentioned here. I have mentioned today injection molding process, extrusion process, blow molding process, compression molding process, injection, stretch, blow molding process, etc. So these are the process through which we can fabricate different plastic products. Yeah. So these are so in a nutshell, in a 3 different sectors I tried to complete the discussion. First, the few about the plastic materials, especially it's from the application point of view, how it is we are using different plastic materials in different sectors. Then second, what are the conditions, what are the criterias need to select for choosing a polymeric material for biomedical applications? What are the checklist? Then what are the standards? One standard it was discussed, what are the standards is available for bio material to be used, what are to fulfill the preconditions for to be used for biomedical applications. And finally, the processing techniques through which we form this type of biomaterial for products which are used for biomedical applications. So hope I cleared some of the I tried to clear some of the topics in your field. So any questions or query, I'll be very happy to answer it.
Thank you
Dr.
very
Devi?
much, sir. Thank you very much for the expert lecture, full with concept of basic theory, processing techniques in details, technological development, and recent application in biomedical domain. Now the session is open for interaction. If anyone has any doubts or any questions, you can interact. You can raise your hand and interact with sir directly. If any participants wants to interact with sir, you can raise your hand.
Yeah, I think I can yeah, I think I am also sharing my. I think Dr. Devi, can you share my email ID or mobile number so that in future also, if you want to clarify anything in especially for the or processing techniques, or even I am also associated with the testing of different products. So accordingly, if you want different type of testing, if you need any help regarding the testing of different plastic products, composite products, we can also discuss. We can also help in that field.
Yes, sir.
So,
I
Dr.
will
Devi,
share the yes, sir. I will share the email and phone number, and all the participants has found it very important and informative for covering the basics of polymers. So details. Thank you very much, sir.
okay, thank you very much, Dr. Devi, for inviting me for this seminar. I'm very glad to be a part of this AICT sponsored Atal FDP program. I thank to all the participants for your kind and very for your kind cooperation during this session. Thank you very much, and thank you to all. Thank you.
Thank you, sir. Mhm. I am sharing the email address and phone number at the chat box. Thank you very much. মোৰ আৱাজটো শুনিছে ন? Thank you very much. Am I audible? Okay. So our next session will start at tomorrow 9:30 a.m. So I'm requesting all of you to be present here on time. Dr. Pradeep Paik sir from IIT BHU will deliver the talk, and the PPTs with due permission from the speakers, we will share it. And for the feedback link, it will be at Atal portal. So after completion of the courses, the feedback can be given by you. And in the valedictory session also, feedback, any interested people can give the feedback. So now you can leave the meeting. Thank you so much. And tomorrow we will meet again at 9:30 a.m. And the attendance will be taken by since we are using WebEx, so attendance will be taken by the software. No need to worry for that. And please be present here on time tomorrow. I hope all of you have had a good day. Thank you. Thank you so much.